Another process that happens with DNA, and it's important for you to know this is different than transcription and translation, but it does involve working with the double helix. This is DNA replication. And before a cell divides, it is going to need to make a copy of all of its genetic information. So that's what DNA replication is. And again, just like with transcription, complementary base pairing allows us to know what the complementary strand that is going to be made, what the sequence will be. So the same thing as the A, T, C, G. We know that A binds with T and C with G and vice versa. And so we can know when the DNA is replicated what strand will be made. So the 4 main steps of DNA replication that you should know. First, the DNA is unwound. It's a DNA helicase that will unwind and actually result in what we call a replication fork, which is essentially just the separation of the 2 strands. And a DNA polymerase will come along and on each strand and synthesize complementary strands. DNA ligase is an enzyme that will connect the segments. So if you can imagine, we have 2 strands that are being copied by a polymerase. In this direction, the complementary strand can just be made continuously. However, on the other strand, there's a gap in the replication. Only parts can be made at a time. Only certain parts or certain numbers can be made at a time. So you end up with just these bits of code or sequence. And so DNA ligase comes in and helps bind those parts or segments so that you have one single strand. And so we call this semi-conservative replication because each new strand of DNA contains parental DNA and newly synthesized DNA. Additionally, once these new strands are made and we have a new two double helices, you're going to have new histones be made that will help organize that DNA. So new histones are synthesized and it helps to organize those new DNA strands into nucleosomes and chromatin.